My name is Oscar Dillon. This is my presentation on transcranial direct current stimulation, or tDCS for short, and its application on chronic pain. tDCS consists of a small electrical stimulation device that uses two pads of opposing charges to either excite or inhibit neurons in the brain. Simply put, it gives a targeted part or parts of the brain a nudge to change its activity. tDCS applies low-level electrical current to the scalp, influencing neuron membrane potential. It does so through an anodal or a positive charge, depolarizing neurons, making them more likely to fire, or a cathodal or negative charge, which hyperpolarizes neurons, reducing the likelihood of firing. In doing so, it may influence synaptic transmission, influence neuroplasticity, cause changes in the brain circuits and networks and how they communicate, or may cause hemodynamic changes. Lorimer Moseley describes chronic pain as a protective output of the brain. Further, he states the brain can produce pain when it perceives the body is under threat. Unlike acute pain, which is the body's immediate response to threat, chronic pain usually persists after threat and is considered chronic when it persists for 3 to 6 months. The activation of specific networks in the brain associated with chronic pain gives way for the hypothesis that the modulation of neuronal activity could be a potential treatment. Several studies have evidenced tDCS as a promising, non-invasive technique for the treatment of chronic pain. In 2006, Philippe Fregny and others found a significant reduction in pain associated with fibromyalgia. In 2010, Andrea Antal and others showed that anodal tDCS could alleviate chronic pain and modulate cortical excitability. In 2012, Kirsten Ludke and others found positive results in combining tDCS and cognitive behavioral methods in managing chronic lower back pain. And in 2014, Vesegi and others found tDCS to have a modulatory effect on sensory perception and pain. Now, these are all promising results. However, making conclusive judgments on tDCS is made difficult due to the different measures and study designs used in these experiments. For the most part, this pertains to no universally agreed upon method of application. For example, the location of electrodes, the intensity or duration of stimulation, or the number or frequency of sessions. In sum, the lack of long-term data and placebo effects make assessment hard. However, its non-invasive nature and minimal side effects offer opportunity for further study. There are many physiological mechanisms of tDCS that could affect chronic pain. Firstly, neuronal excitability modulation. By altering the balance of excitation and inhibition, we could influence the perception and processing of pain. Secondly, synaptic plasticity. It could change the strength in which synaptic signals are sent, which could alter pain perception. This could happen in the form of long-term potentiation or long-term depression. Third, it may influence changes in endogenous pain control systems by modulating the release of neurotransmitters and endorphins, which could change the perception of pain. Fourth, it may reorganize the maladaptive neural pathways associated with chronic pain and mitigate negative adaptations. Also, it may alter functional connectivity between brain regions, resulting in changes in communication between networks and altering pain perception and processing. It may modulate pain threshold, making it harder to perceive pain. Or lastly, it could cause thalamic modulation, which could influence transmission of pain signals to the cortex, altering pain perception. There are several limitations that persist of the research done on tDCS for chronic pain. These pertain mostly to methodological variation across the studies, including differences in stimulation parameters and targeted brain regions. A lack of standardized protocols can lead to inconsistent application, making outcomes less reliable. Distinguishing genuine therapeutic effects from placebo is challenging due to tDCS's minimal side effects. Additionally, many studies focus on short-term outcomes, leaving questions about long-term efficacy and safety. Furthermore, individual differences such as the cause of pain and brain anatomy and physiology can influence results, but these factors are not always accounted for in the research. Based on the former limitations, to enhance our understanding of tDCS's efficacy in treating chronic pain, future research should prioritize establishing standardized protocols. This would address the current methodological inconsistencies and improve the reliability of outcomes. There's a pressing need for long-term studies to ascertain both the sustained efficacy and safety of tDCS over extended periods. Investigations should also incorporate control measures to discern genuine therapeutic effects from placebo responses. Moreover, given the significant individual differences observed in response to tDCS, studies should take into account variables like the underlying cause of pain and individual brain anatomy and individual physiology to ensure a more personalized and effective treatment strategy. Thank you.